Stuart Newman, Professor of Cell Biology and Anatomy at New York Medical College and a founding member of the Council for Responsible Genetics in Cambridge, Massachusetts, is one of the many prestigious scientists worldwide to express strong opposition to these contentious experiments. He was interviewed recently by a member of the Italian National Bioethics Committee, Dr. Assuntina Moresi, herself a scientist, and who also argues robustly against the proposals. The following is an excerpt from that interview and underlines why this research should not be allowed to go ahead.
I'm Stuart Newman. I'm a developmental biologist at New York Medical College in New York, and I was a founding member of the Council for Responsible Genetics, and I've been interested in the applications of developmental biology to humans. What I think, and what my colleagues who I've spoken to about it think, is that it's kind of a funny way to make humans. It's not simply the three-parent aspect of it, but it's a very kind of disruptive process by which fragments of cells from two different women, the nucleus of one egg and the cytoplasm of another egg, are kind of ripped apart and put back together again and then fertilized, and it's very experimental. We don't know what the effect on the full person would be of this kind of experiment. So it's a very strange process, and anybody who thinks that it's just a routine type of thing really doesn't understand the science of it.
Professor Newman was then questioned about the safety of these procedures.
It's, every time you do an experiment like this and you try to transfer it into another species, like if you do it on rabbits and then you try to do it on mice, or you do it on chickens and then you try to do it on frogs, things always come out different. And then you have to do a whole kind of cycle of experimentation with many negative outcomes before you get the conditions right for it to work in the new species. So here, based on things that have been done in mice and rats, the HFEA is proposing to allow it to be tried in humans, and it's just an experiment when you do it in humans. And so what happens if instead of getting an improved person, you get a person that is impaired from this procedure? Do you discard the experimental outcome? What do you do?
Professor Newman was then asked whether these procedures constituted modification of the germline.
The human organism has two kinds of genes. There are genes in the cell nucleus, and there are genes in the mitochondria. And the mitochondrial transfer procedures are proposed because some women have mitochondria that have mutations that will impair the health of their offspring. People talk about germline modification as modification of the nuclear genes. And they say, well, the mitochondrial transfer doesn't affect the nuclear genes, and therefore it's not germline modification. But that's not exactly true. But from the point of view of the embryo, the developing embryo, that embryo is made up of part of the cell of one woman, part of the cell of the other woman. That embryo is the product of germline engineering. There's no getting away from it. So it's just only because of some legal convention that the woman who is contracting for the procedure doesn't have her germline modified that they call it not germline modification. There's an idea that a lot of people believe, but most biologists don't believe anymore, that the nuclear genes have some kind of program or blueprint for producing the organism. And so if you have some mixture, part of this cell, part of that cell, as long as it has some intact nuclear genome present, then according to this false idea, the blueprint is there, the program is there, so there's nothing to worry about, because the whole blueprint is intact. But that's not the way it works. The genes are interacting with the cytoplasm, with the environment, and even when the germline is established, the germ cells are established in the development of the embryo, the mitochondria participate in that establishment through interactions. If the mitochondria are heteroplasmic, then the germline might be affected, or at least the germ cells will be affected. So you'll have inheritable defects, not because the genes of the nucleus have been affected, but because the gametes, the sperm or the egg that are developing in that embryo, are impaired because of the procedure that was done. So this is very complicated, and it can't be reduced to the idea of a genetic program. But what you're doing there is you're manipulating the very materials of what it is to be human.
Professor Newman was asked to comment on the concerns of other professionals in this area.
What about your colleagues?
Any developmental biologist that I speak to about it says that, you know, of course this is not appropriate to use on humans. Some people say, well, maybe, you know, 25 years down the line we could try it on humans. And so then I say, well, what would be the procedure to get from here to there? Are we going to do experiments on humans in order to make this technique acceptable to use in a routine fashion? And they say, well, we can't do those kinds of experiments on humans. So ultimately, people come to the conclusion that there's really no way to get, no ethical way to get from here to there. And it seems to me that what has, you know, what is being considered by the British HFEA is a kind of permission to do experiments on people, so that maybe down the line, with all the errors that occur and everything like that, the typical person will be comfortable using it. You know, it seems very unethical to me. I can understand why people would want to have healthy children. Everybody would. But these procedures that are proposed are experimental procedures that it seems fairly likely in many cases to do more harm than good. So in some cases it might alleviate the mitochondrial condition. In other cases it will introduce new problems. I don't want to be understood as being anti-genetics or anti-technology. I use genetic techniques in my own laboratory research on chicken embryos and mouse embryos. So I think these are very powerful techniques, and they will have benefits for human health. But we're really just at the beginning of, and I think that they should never be applied to making new kinds of humans.